Picture this for a second. Your brain is essentially a highly advanced, ultra-high-definition projector. And most of the time, it's taking a live feed straight from the outside world, through your eyes, and broadcasting that right into your conscious mind. But what actually happens when someone comes along, flips a switch, completely unplugs that live feed, and plugs in an old, fragmented hard drive filled with your deepest memories instead? Well, today in this explainer, we're unpacking a literally mind-bending breakthrough in neuroscience that explains exactly how that switch gets flipped. Seriously, what if your brain could just shut off your eyes and completely replace reality with your own memories? I know, it sounds exactly like the plot of a sci-fi movie, but it's actually the core biological mechanism behind one of humanity's most profound and honestly, historically misunderstood experiences, the psychedelic hallucination. For literally thousands of years, people have experienced these incredibly vivid, self-generated worlds without having any real idea what mechanical levers were being pulled inside their own heads. Well, until now. Okay, let's dive into this. A truly fascinating new study has finally managed to map out the exact neural circuitry of hallucination. Researchers have essentially figured out how psychedelics hijack the brain's visual processing centers, basically forcing the mind to dream while you're completely wide awake. But to really grasp how we finally cracked this biological code, we kind of need to look back at just how long we've been trying to solve this mystery. Ancient roots to modern science, tracing the millennia-old mystery. You see, our fascination with altered states absolutely isn't some new modern trend. We're talking thousands of years ago. Ancient indigenous cultures were already using these compounds. The Aztecs used psilocybin mushrooms for medicine. Cults down in the Andes were consuming mescaline-rich San Pedro cacti. Archaeologists have literally found 5,000-year-old peyote buttons in Texas, and even a ritual bundle laced with DMT stashed in a Bolivian cave. So for millennia, these were viewed as purely mystical experiences. But the modern scientific journey, that really kicks off in 1938, when a Swiss chemist named Albert Hofmann synthesizes LSD in a lab. And that specific moment sets the stage for the massive breakthroughs of the 1970s and 80s, which is when researchers finally found the biological lock that all these chemical keys were fitting into, a specific brain receptor called 5-HT2A. Which brings us to the neuroscience of tripping and unpacking the biological mechanism. Let's take a closer look at this 5-HT2A receptor. I want you to think of this thing as a highly sensitive antenna sitting right there on the surface of your brain cells. It is a really crucial part of your brain's serotonin system, and serotonin is a massive player in how you feel on a daily basis. It heavily affects your mood, and it deeply influences conditions like anxiety and depression. But when psychedelics bind to this specific 5-HT2A antenna, it doesn't just tweak your mood, no. It triggers intense visual hallucinations. It drastically alters your entire perception of reality. And that right there leads scientists to a massive, game-changing question about how we treat mental health. So the crucial point is really neuroplasticity. That's the ability of your brain cells to literally rewire themselves and start communicating in entirely new ways. A lot of modern scientists strongly believe that the true therapeutic benefit of psychedelics, the real reason they show such incredible promise for treating severe depression and PTSD, well, it isn't necessarily the spiritual trip itself. It's actually this underlying neuroplasticity. The drug essentially forces the brain out of its rigid, depressed ruts, allowing it to forge fresh, healthy connections. Which brings us to the literal, multi-billion-dollar question that's currently driving modern pharmacology. Is the mystical visual trip absolutely required to treat depression? Or are the hallucinations merely a side effect of that neuroplasticity? Because think about it. If the hallucination is just a side effect, then we could, theoretically at least, design drugs that heal the brain without sending the patient on a massive 12-hour mind-bending visual journey. But to strip the hallucination away, we first have to figure out exactly how the hallucination is generated in the first place. And that takes us inside the mouse experiment, visualizing glowing brain cells. To actually map a hallucination, researchers turned to a super sophisticated mouse model that honestly just shows how incredibly advanced our neurological tools have become. Step 1, they genetically engineered mice so that their brain cells literally glow when they become active. Basically, the harder the cell works, the brighter the glow. Step 2, they showed these mice simple visual stimuli. We're talking moving black and white bars or just blank screens, just to establish a solid baseline of how their normal visual cortex operates. Step 3, they injected the mice with a really potent chemical that selectively activates that exact same 5-HT2A receptor we were just talking about. And finally, Step 4, using some brilliant technology developed by lead researcher Thomas Knoefel, they recorded the precise increases and decreases in voltage across the surface of the brain. They were literally watching the brain's electrical grid in real time. Now for the big reveal. Unlocking the waking dream, the aha moment. And this brilliantly illustrates what happens next, because the results were incredibly stark. Before the drug, the mouse's primary visual cortex is just humming along at a steady 5 Hz oscillation. It's doing its normal, everyday job, processing the external visual stimuli coming right in through the eyes. But man, after that drug hits the receptor, everything changes. The brain suddenly shifts into these intense, slow, rhythmic waves known as theta rhythms. Now, theta rhythms are typically linked to things like attention and memory. But the real aha moment here was seeing exactly where these rhythms connected. The visual processing area of the brain completely synchronized with the retrosplenial cortex. And that is a completely different region of the brain, entirely responsible for encoding, storing, and retrieving memories. 18 milliseconds? That is the exact delay the researchers measured in this massive synchronization. What that specific number actually represents is a literal traveling wave of electrical activity firing back and forth, completely bridging the gap between the brain's visual center and its deep memory storage banks. Let's break down exactly what that 18 millisecond wave is actually doing in there. Basically, the psychedelic acts like a master switchboard operator. First, it deliberately dampens the brain's response to what the eyes are actually seeing. It essentially turns down the volume on the outside world. Second, it aggressively boosts the connections to those retrosplenial memory areas. And third, and this is wild, because the brain absolutely hates a vacuum, it begins to fill in the missing visual information by inserting fragments from its own internal memory banks. The brain literally stops looking out the window and starts looking in the mirror. It takes those internal memories and projects them straight onto the external visual field. Lead researcher Dirk Jenke described the phenomenon absolutely perfectly. He said it's remarkably similar to partial dreaming. Under the influence of the drug, the brain's internal imagery overrides external reality, creating a vivid, self-generated world. That is incredibly profound when you take a second to think about it. The waking dream of a hallucination isn't just the brain inventing random things out of thin air. It's the brain aggressively surfacing its own archived data and feeding it directly into the visual cortex, all while you are wide awake. This brings us to the future of therapeutics and stripping the hallucination from the healing. Now, what's really interesting about this slide is how it grounds the science. Because to be totally rigorous, we've got to acknowledge the limitations of a study like this. First off, while mouse and human brains definitely share fundamental features, they're definitely not a perfect one-to-one map. We obviously can't say with 100% certainty that human hallucinations mirror this exact 18 millisecond circuit down to the very last detail. Secondly, the authors themselves point out a much simpler variable. The mice might have just gotten a bit distracted by looking at those repetitive moving bars, and that alone could cause their brain activity to shift inward. Science is all about objectively observing the data, and these are super important caveats to keep in mind as the research moves forward. But even with those caveats, mapping this circuitry is a monumental step forward. By successfully identifying the exact switchboard mechanism that swaps external reality for internal memory, scientists are closer than ever to isolating the hallucination from the neuroplasticity, which really leaves us with this final provocative question. Will decoding this biological projector finally unlock the ultimate hallucination-free cure for mental health? If we can somehow engineer a drug that triggers the exact same massive healing neuroplasticity, rewiring the brain completely out of depression and trauma, without flipping the switch that causes a vivid 12-hour waking dream, well, that would permanently revolutionize modern psychiatry. It's an absolutely thrilling frontier, and we can't wait to see where it goes next.